en varios departamentos, de cargo de profesor en varios departamentos. Bueno, Igal se especializa en temas de biofísica y materia blanda. Hoy nos va a hablar sobre cromatina. Y bueno, como siempre que lo presento, me gusta resaltar de Igal que su grupo siempre está abierto a los argentinos, fuimos muchos los estudiantes que pasamos por ahí, siempre el grupo de Igal, siempre hubo un mate sobre la mesa, a veces también nos de sabana. Y también en eso, Igal siempre mantuvo lazos muy fuertes de colaboración con Argentina, es doctor honoris causa de la UBA, es miembro de la Academia de Ciencias de Buenos Aires, fue muchas veces, vino muchas veces con el programa de Einstein, así que bueno, eso es lo que quería estar. Bueno, Igal, te dejo.
Gracias. Buenas tardes. Tuve una CB, tengo afasia, tengo dificultad para hablar. Por favor, tengan paciencia. Gracias. Chromatin structure. Marcelo Carignano and Martin Kruger work on the theory part. No. Chromatin refers to a mixture of DNA and proteins that form the chromosomes founding the cells of humans and other higher organisms. Chromatin has many components. At the small scale, chromatin is double-stranded DNA. DNA together with histone octamers, such as proteins, form nucleosomes. Nucleosomes could be thought as bits in a linear polymer model. The polymer forms to form an irregular fiber. Further heterogeneous folding form the chromosomes. Physical genomics. How is chromatin structure how is chromatin structure organized? How does chromatin structure change due to external and internal cues? How does chromatin structure influence transcription? In this talk, I will concentrate on the first question. The understanding of polymer physics is important to approach the problem of chromatin organization. However, chromatin does not behave as any of the standard models. The chromatin is a very special polymer and needs a different theoretical treatment. The self-returning excluded volume model is an extension of the self-returning random walk model developed by us and published in 2020. The SRW is a random walk with the possibility to return over itself. Each new step in the SRW could be a return step or a forward step according to a probability distribution function. The probability distribution function depends on one parameter, alpha. The SRW has two problems. it has not excluded volume, and it is not a linear chain. We remove these problems in the SR excluded volume model. The final SR excluded volume configuration consists of a linear polymer with clusters, but with no overlaps. We use different folding parameter alpha and 4 different global volume fraction phi. Then in total, we have 12 simulation cases. We choose the units so that each bit represents a nucleosome. Notice that alpha is a parameter in the simulation. Locally, there are variations in the volume fraction, as the resulting configuration are not homogeneous. For the more dense case, we model the model chromatin has the size of human chromosome 16. It is very difficult to visualize in 3D. Using a cross-screening method is helpful. Here we wrap the model chromatin with a mesh created with the alpha-shape method. Looking at the entire structure of the mesh, we see that it is reminiscent of the interface between two quite continuous spaces. SR excluded volume is consistent with chromatin as a non-crystalline condensate. The formation of the correlate structure of chromatin is the result of the two probability distribution functions and excluded volume that effectively capture the processes leading to the packing of chromatin. ChromStem experimental procedure. Here we show how the output from ChromStem experiments are treated in order to find how chromatin organizes and identifies packing domains. Starting from a stack of images, domain centers are determined and a 2D mass integral is calculated. The domain radial are measured from the domain density profile, and the parameter the packing domain D parameter is measured from the mass integral, or the experimental procedure. This is only. This is not the same thing. They are cut. This and this is not the same thing. The SR excluded volume model is for a single chromosome, while the experiment corresponds to the whole nucleus. There is a very good agreement between the theory and the experimental ChromStem. This and this is the same thing. pioneering plots is a way to visualize statistical data commonly used in biological studies. The shape of the plot represents the distribution of or frequency distribution. The mean value and the mean value and standard deviation are displayed with black lines. Grouping the results by alpha, same color, same color, we see that the distributions shift to larger radius as the overall volume fraction increases. The majority of the domains have the radius between 50 and 100 nanometers approximately, but the distribution is highly skewed towards larger radius. There is very good agreement between the theory and the experimental results. R is our excluded volume, experimental R is the same thing. Calculating the individual D for each configuration, we can characterize the distribution of D values over the 12 different ensembles. The mean value of the distributions increase with increasing volume fraction and have a small dependency on alpha. There is a very good agreement between the theory and the experimental results. The theory, experimental V is the same thing. We begin with a single 3D chromatin configuration generated by the SR excluded volume model, consisting of 500,000 bits, approximately the size of a human chromosome 15. To visualize its internal structure, we sweep through it with a 50 nanometer slick slab, mimicking the type of 2D imaging implemented in ChromStem. Each frame of this video could correspond to the actual ChromStem experiment. A single slice through a single cell, and as the experiment, what is seen depends entirely on the depth and position of that slice. The video reveals how local compaction and anisotropy emerge naturally in the SR excluded volume model, even without architectural proteins or loop exclusion. Domains are not spherical or uniformly distributed. They appear elongated, layered, and irregular, consistent with non-random folding. This supports a model in which chromatin architecture, chromatin architecture, chromatin structure is emergent and heterogeneous with coordination number as a proxy as a physical proxy for accessibility. Coordination number serves as a proxy for chromatin accessibility, linking structure with potential function. Here we show a schematic representation of a polymer with architectural proteins coesine and CTCF. This is not equal than this. This is not this and this, and is different. Wait. In this slide, we prove single-cell variability of the anchor chromatin loop with the SR excluded volume model. What you are seeing in each panel is a small part of 4 chromosome scale SR excluded volume configuration. Configuration length is 20 megabase pairs, but the part shown here is 360 kilobase pairs from a loop. The loop starts and ends at the same genomic coordination defined by the same pairs of convergent CTCF protein sites. Green series is the same. Coesine proteins overlap with CTCF proteins in this case. Chromatin is colored by coordination number. Red indicates dense compaction regions. Blue is more open, and white highlights the surface of packing domains. The rotating views show that folding and compaction that folding and compact geometry differs substantially across cells despite fixed anchors. Looping in 3D is not deterministic. Hi-C is an experimental technique to reveal contacts in the chromatin DNA. The results are displayed as 2D contact map with the color reflecting the contact frequency. Single cell contact maps are limited and do not immediately reflect any pattern. But ensemble average 1 million cells reveal an organized pattern. Squares features of enhanced contacts along the diagonal of the map are called topological associating domains, or TAD. We think of TADs as a relatively weak perturbation affecting SR excluded volume configuration. I'm sorry. In this slide, we connect the 3D configurations shown to slide back to their corresponding 2D contact maps. Bottom. Similar to what is captured in single-cell high-C experiments. Each 3D configuration includes in its mean coordination number a rough measure of local packing density. All maps show, all maps cover the same genomic region with the green dash box marking the touch boundaries observed in the ensemble average. We will talk about this in a few slides. Despite sharing the same loop anchors, these contact patterns are strikingly different. Some maps show dense contact clusters, others are limited and fragmented, especially in the cells with lower coordination number. The red arrow on the right highlights this point. The most open structure with coordination number about 4 leads to a contact map with minimal interaction structure. Oops. These examples illustrate that loop anchoring does not guarantee consistent spatial holding. Instead, there is substantial cell-to-cell variability, both in 3D configuration and in the 2D measurable contacts. This supports the view that chromatin folding is probabilistic, not deterministic. That single-cell data reveal a rich, more heterogeneous landscape than ensemble average alone. This slide explores how the length of an just a minute. I'm sorry. This slide explore how the length of an internal loop influences the 3D chromatin structure in the SR excluded volume model. All configurations here represent the same chromatin sequence of 360 kilobase pairs anchored by convergent CTCF site shown as green spheres. This is not the same thing. This, and this, etc. What varies is the length of an internal loop formed by coinciding proteins, yellow spheres. This. Which progressively decreasing from left to right, from 260 kilobase per to 108 kilobase per. As we reduce the loop length, we observe only a modest effect of compaction. The structure still exhibits a high degree of variability, both in geometry and how the domains are formed. Domains are arranged. The videos illustrate that shorter loops do not necessarily impose stronger constraints. The global folding remains flexible and heterogeneous, even under loop exclusion like conditions. On this slide, next slide, we will look at how this structure translates into contact maps and how little consistency remains even with loop anchors present. Here we examine the 2D contact maps corresponding to the structure from the previous slide. Each ROM shows a 3D structure on the top and its single cell has a single cell 2D contact map below. The internal loop is again formed by coinciding proteins, yellow spheres, with length decreasing from left to right. The green dash box outlines the full 360 kilobase pairs, while the yellow box marks the internal loop segment. As the loop shortens, we see a modest reduction in coordination number. Compaction decreases slightly. But even more striking is how the contact patterns change. Some maps show dense clusters and structural domains, while others appear scattered and disordered, especially with short loops. This and this is not the same thing. This highlights two key points. First, loop exclusion alone does not ensure a compact or stable structure. Second, the contact map is sensitive not just to loop anchor position, but the full 3D context. This supports these results support the idea that chromatin architecture is a variable and probabilistic, even with loop anchors are present. This slide summarizes how loop exclusion by architectural protect, a process happening on 3D space, is sought to generate the key features observed in high-C maps. In each schematic, CTCF binding sites are shown as red spot, and coefficient complexes as green block complexes as blue rings. CTCF acts as a blocker or stopper that holds the progression of coefficient during loop exclusion. As coefficient translocate along the chromatin, the loop it excluded grows, reflecting the increasing genomic distance between its two bond sites. When the exclusion holds between the convergent CTCF sites, it creates a loop structure, and depending on where the line anchor lies, we observe distinct signals in the high-C matrix. When exclusion is halted by both convergent CTCF stoppers, it stabilizes a loop, and in the 2D contact map, this creates a characteristic corner spot of a tile. If only one side of Quaking block is blocked by TCTF, while the other side continues to extrude and holds at a more random internal location, the result is a horizontal stripe, a zone of enhanced contacts along a extending downstream from a anchor. Conversely, when Quaking holds first on the inside internal sides and continues extruding on the opposite slant until it reaches a distal CTCF side. The map displays a vertical stripe reflecting upstream contact enrichment. Finally, when Quaking stalls somewhere within the domain between two anchors or inside a loop, it contributes to the interior of the touch square. These internal stalls reinforce the dense contact matrix inside domains, but do not produce sharp order features. Together, these scenarios explain the corner sides and interior enrichments that define the touch triangle seen in ensemble-averaged ensemble IC maps. But crucially, these features only emerge after averaging over many cells. This raises an important point. Touch are statistical domains, not fixed structures. They reflect a weak organization influence, not a deterministic folding blueprint. We look at the ensemble average to the contact map built from 40,000 3D self-returning excluded volume configurations. Each configuration contains either a full or partial loop of 360 or 60, 70 and 20 kilobase pairs formed through a loop extrusion-like procedure. The contact map uses a logarithmic scale to highlight enriched interactions. What emerges is a familiar pattern that, like squares along the diagonal, with depletion interaction between domains. These features arise only after averaging over many heterogeneous cells, heterogeneous structures. In individual configurations, loop and constraints appear inconsistently, but when pulled, they reveal the stereotypical touch pattern. This shows how ensemble-averaged can turn structural noise into statistical signals, summarizing what you see what we see in population high C data. It underscores the key point. That are statistical constructs, not physical compartments present in every cell, but patterns are that emerge through bias folding across many configurations. The slide connects the structural packing with touch boundaries, using the coordination number as a result. The red line shows ensemble-averaged coordination number along the genome region and the dashboard. Blue line marks the average coordination number across full chromosome. What stands out is the sharp coordination number peaks, which align with the boundaries of touch as shown in the inset contact map. This, and this. These peaks represent a statistical local high density marked by the selective location of CTCF. It is another piece of evidence supporting the view that touch are weak statistical domains. The touch boundaries reflect the strict positioning of the CTCF architectural proteins, which are stop sign for cohesion. Conclusion. The SR excluded volume model offers a scalar phase-based approach to modeling chromatin structure, generating full 3D configurations without relying on detailed interaction networks. This simplicity enables large-scale simulations while still reproducing features seen across diverse experimental platforms, from single-cell high-C to from STEM and chipset profiles. What emerged from this work is a view of genomic architecture as a probabilistic system. Touch and loops are not rigid features. but statistical patterns shaped by weak constraints like loop anchors. Insufficiently contacts in single cell data reveal architectural variability. With its simple and minimal assumptions, the SR excluded volume provides a powerful platform for exploring how genome architecture how genome structure and regulation are physically linked. Thank you very much.
Bueno, podemos abrir las preguntas. Pueden preguntar en castellano, obviamente, si quieren.
Clinical physics.
Yo tengo una pregunta. ¿Cuántos tipos de configuraciones encontrás bajo tus simulaciones? Son
Millón. Number. No. Es 20 megabase pairs. Es un montón. un montón. Es difícil. Muchos. El computer no sabe nada. Es impresionante.
O sea, obtenés más de las que en realidad son posibles en un tipo. Vos, para hacer un experimento de High-C, usás 10 millones de células.
Sí.
Y reproducís un patrón que después vos reproducís con las simulaciones, pero obviamente a lo sumo tenés 10 millones de configuraciones.
Sí. 1 millón. Más o menos. ¿Qué pasa? High-C is irrelevante. Wow. Es el eso. Esto es
un artefacto.
Sí, no. Bueno. Sí, ensemble-averaged, nada más. Pero single cell es diferente. Es diferente, pero es diferente. Y nosotros trabajamos experimentalmente. Vadim. Y yo critiqué, pero Vadim esto es fabuloso, pero esto sí, pero Polymer es, por ejemplo, 8 proteins núcleos ups. Esto 1, 2, 3, 4, 5, 6, 7 proteins no o sí por tu proteins no va o si va. Por ejemplo, pulmón y muscles es diferente, pero madre.
¿Células madres?
¡Qué emocionante! Células madres es bárbaro, pero es diferente. Es un quilombo. Pero se puede.
Sí, gracias.
Sí, muy lindo trabajo, Diego, felicitaciones. En la transparencia anterior vos mostraste una estructura bicontinua y dijiste que estas cosas forman la otra fase es agua.
Sí.
Y en las simulaciones el agua está incluida entonces.
Esto, ¿entendés? No.
Sí, se ve mejor en el anterior, creo, ¿no? Ahí.
Anda.
O sea, el agua forma parte del sistema.
Esto pero es lo de pelote, volvimos, es three-dimensional, es
Complicado.
complicado.
Una muy cortita, muy técnica. Los gráficos estos de violín, ¿qué gráfico es exactamente el eje X? Sí, el eje Y es el tamaño de...
Phi.
Es la dispersión en...
No. Volume fraction.
En la fracción en volumen.
Sí.
Ok, no es un label, tenés algunos arriba de 0.16, algunos abajo.
Esto es diferente. Y bárbaro. Y después
Sí. Pero en este
No,
gráfico,
depende.
en ese gráfico, ¿qué es el eje X? En este gráfico.
No sé.
Número de observaciones.
No. Esto es irrelevante. Esto, sí.
Ok.
Esto es cells de...
Un histograma, básicamente.
Gracias. Un hierro.
Bueno, hacemos ahí otro.